Hello students, today's topic is synthesis of purine nucleotides, inosine monophosphate, adenosine monophosphate and guanosine monophosphate. So purine nucleotides are synthesized in almost all the cells, but mainly in the liver, and it occurs in the cytoplasm of the cell. So there are two ways by which the purine nucleotides are synthesized. Number one is de novo synthesis. And number two is the salvage pathway. So de novo synthesis, de novo means new or starting from the beginning, when it is synthesized from the starting materials like amino acids or bicarbonate. And salvage pathway means when purine bases released by the hydrolytic degradation of the nucleic acids and nucleotides are salvaged and recycled. So first, de novo synthesis. First I will show you the sources of atoms of purine ring. So purine nucleotide. So purine ring is formed by fusion of pyrimidine ring and imidazole ring. So in the pyrimidine ring, the atoms are numbered in anti-clockwise direction: 1, 2, 3, 4, 5, 6. Here clockwise direction: 7, 8, 9. Now this N1 is the source is aspartate. C2 the source is N10 formyl tetrahydrofolic acid. The source of N3 and N9 is glutamine. The source of C8 is N5 and N10 methynyl tetrahydrofolic acid. Source of N7, C4 and C5 is glycine, and source of C6 is the respiratory carbon dioxide. Now first IMP is formed, and from IMP, AMP and GMP are formed. Now we will go through the reactions of purine synthesis. First, there is the preparatory phase or preparatory step or step 0, where the building material PRPP is synthesized. So we get ribose 5-phosphate from the HMP shunt. It is converted into 5-phospho-ribosyl 1-pyrophosphate by the action of the enzyme. It is also known as PRPP, and the enzyme catalyzing this reaction is PRPP synthetase. ATP is required. Now step 1 starts from the next step, and in de novo synthesis this PRPP provides the foundation on which the other atoms are added to form the purine nucleotide. First, this PRPP is acted upon by the enzyme glutamine PRPP amido transferase, where glutamine donates the amino group and converts into glutamate and inorganic pyrophosphate is released, and the product is 5-phosphorybosyl 1-amine. So the amino group replaces the inorganic pyrophosphate, and it contributes N9 of the purine ring, and this is the rate limiting or committed step of purine synthesis. Next, this 5-phosphoribosyl 1-amine is converted into glycinamide ribonucleotide. Glycinamide ribonucleotide or GAR by the enzyme GAR synthetase. Here, glycine provides the C4, C5 and N7. C4, C5 and N7 of the purine ring. So glycine combines with this 5-phosphoribosyl 1-amine to form GAR. Here ATP is required. Now this GAR is converted into formyl glycinamide ribonucleotide or FGAR by transfer of the formyl group from N5 N10 methinyl tetrahydrofolic acid. And the enzyme is GAR transformylase. GAR transformylase. And here the C8 atom is added in this step. Now this FGAR is converted into formyl glycinamidine ribonucleotide or FGAM by the action of the enzyme FGAR amido transferase, where there is transfer of the amino group from glutamine to FGAR and it forms glutamate. ATP is required. Okay, so in this step glutamine provides the N3 of the purine ring. Now this FGAM is converted into 5-amino imidazole ribonucleotide. 5-amino imidazole ribonucleotide or AIR by FGAM cyclase. So there is cyclization of the structure. Here also ATP is required. Now this AIR is carboxylated by carboxylase enzyme to form carboxy amino imidazole ribonucleotide or CAIR. So this carbon dioxide, it provides the C6 of the purine ring. Now this CAIR is acted upon by the enzyme Sychar synthetase to form Sychar or N-succinyl 5-amino imidazole carboxamide ribonucleotide or Sychar. So, here ATP is required and aspartate is required. Aspartate provides the N1 of purine ring. Aspartate is required and ATP is required. ATP is converted into ADP and inorganic phosphate. So this Sychar is acted upon by the enzyme Sychar lyase, where fumarate is released and it forms 5-amino imidazole 4-carboxamide ribonucleotide or ICAR. Now this ICAR is converted into 5-form-amino imidazole 4-carboxamide ribonucleotide or FICAR. Okay, so here in this step N10 formyl tetrahydrofolate donates the formyl group and it is catalyzed by the enzyme ICAR transformylase, and this N10 formyl tetrahydrofolic acid contributes the C2 of the purine ring. Now this FICAR is converted into IMP or inosine monophosphate by the enzyme IMP synthase. Water is eliminated. So this is the first purine nucleotide which is formed, and from IMP we get AMP and GMP. So IMP is acted upon by the enzyme adenylosuccinate synthetase. When it combines with aspartate, GTP is required. It forms adenylosuccinate, and adenylosuccinate is converted into AMP by the enzyme adenylosuccinase. Fumarate is released, so AMP is formed. Then XMP is formed from IMP by the action of the enzyme IMP dehydrogenase. NAD is required. Then XMP is converted into GMP by trans-amidinase. Amidinase or amidotransferase. Here, amino group is transferred from glutamine to XMP to form GMP, and glutamine is converted into glutamate, and here ATP is required, which is hydrolyzed into AMP and inorganic pyrophosphate. So that is how the purine nucleotides are synthesized by the de novo synthesis. Now the salvage pathway. So in the salvage pathway, the free purine bases like adenine combines with phosphoribosyl pyrophosphate to form AMP and inorganic pyrophosphate. By the action of the enzyme, adenine phosphoribosyl transferase or APRTase. Likewise, guanine combines with PRPP to form GMP and inorganic pyrophosphate by the action of the enzyme hypoxanthine guanine phosphoribosyl transferase or SGPRTase. Similarly, hypoxanthine combines with PRPP to form IMP and inorganic pyrophosphate, and the same enzyme HGPRTase is required. This is the salvage pathway. Now the regulation of synthesis of purine nucleotides, it is mainly by the feedback inhibition. So we know that from ribose 5-phosphate we get PRPP. From PRPP we get phosphoribosyl amine. From phosphoribosyl amine we get IMP. From IMP we get adenylosuccinate. From adenylosuccinate we get AMP. IMP we get XMP and then GMP. Now the products of these reactions, the end product IMP, AMP and GMP, they inhibit this reaction as well as this reaction. So then PRPP forms histidine or pyrimidine nucleotides. When these are in abundant, then there is no need of purine synthesis, so they inhibit these reactions. As well as AMP will inhibit this reaction and GMP inhibits this reaction. So this is by feedback inhibition. So this is about synthesis of purine nucleotides. Thank you.